AS Level Biology 9700, Paper 2, Variant 1, October November 2024, question number 3. And question number 3 is Figure 3.1 is a photomicrograph of a transverse section through a region of the wall of the bronchus in the gas exchange system. And what is bronchus? You know, the rest part of the respiratory tract, trachea, from trachea is divided further into bronchus, two bronchus, and then bronchus further branch or divide into bronchioles, and bronchioles, ends of the bronchioles, alveolus is present, and alveolus where the gas exchange takes place. So here you can see J and K. These are the two labelings inside the gas exchange system bronchus. And here you can see the next question. Identify the tissue J and K shown in figure 3.1 and suggest how the wall of the bronchiole differs from the wall of the bronchus for these two tissues. So first of all, I have to that the J. Why I have written already solved this because uh, it will be saving time. So you can see that J is a cartilage. Cartilage, what is cartilage? Is a firm, flexible connective tissues composed mainly of cartilage cells, which are embedded in a gel-like extracellular matrix, which is rich in collagen. So here you can see the J is cartilage and it is embedded, these cells are embedded into extracellular matrix. So collagen is there, and but you know the bone, just like the bone, it don't have calcium deposits inside, so it is more flexible as comparison to uh, bones. So J is cartilage. And the other labeling K is smooth muscle. It is a smooth muscle. And smooth muscles are composed of smooth muscle cells. They are elongated, spindle shaped. They are elongated, spindle shaped, so capable of contraction. So the function of muscles is contraction and relaxation. Now, the next question, that is the difference between bronchus and bronchiole on the base of for these two types of tissues. That is the cartilage and smooth muscles. They are present in different ratios in both of these bronchus and bronchioles. Already told you that the bronchus is further divided into bronchioles, and at the end of each bronchiole, alveolus is there. So alveolus is the gas exchange surface where the exchange of gases takes place. That is, oxygen is with your inhaled oxygen is taken in and exhaled carbon dioxide is and carbon dioxide is exhaled out. So in this way, the exchange, because outside the alveolus, blood capillaries are there which have more concentration of carbon dioxide as comparison to alveolus, which has more concentration of oxygen. So oxygen is entered into these blood capillaries, and from these blood capillaries, the carbon dioxide is entered into the exhaled air. So in this way, carbon dioxide is exhaled out first of all. So during this process of gas exchange system, that we have to tell, that is the difference. Bronchus contains cartilage. Bronchus contain cartilage because it has to bear the more pressure as air is entered through this bronchus into the bronchioles. So the cartilage is flexible, so it will perform structural support and keep the airways open, due to which, due to its flexibility, due to whether it is there is high pressure, so the it will remain open. So bronchioles lack cartilage but contain thick layers of smooth muscles. As you only, the bronchioles don't have cartilage, but they have thick layers of smooth muscles. It will also protect these muscles. So bronchus contains less smooth muscles as comparison to bronchioles. Bronchus also contains smooth muscles, but as comparison to bronchioles, the uh, percentage, or you can say the ratio of these smooth muscles will be less in the bronchus because it has more cartilage inside and bronchioles lack that cartilage. So in this way, because they have to face the difference in the pressure during the gas exchange process. Now, next question is, tuberculosis is an infectious disease. Is an infectious disease that affects the human gas exchange system. The pathogens that causes TB secrete a protein that can be detected in saliva. So the protein which is secreted by the tuberculosis. So early diagnosis of TB is important in reducing the transmission of the pathogen. Scientists have developed a test strip for TB that uses monoclonal antibodies, artificially made antibodies for TB. So monoclonal antibodies are specific in their function. As you know, the antibodies have the specific shapes or with which only the specific antigens can bind. So these test strips contain mobile monoclonal antibodies that bind to one part of the protein secreted by the pathogen, immobilized monoclonal antibodies. Two types of antibodies will be used over here for this investigation. That is the mobile monoclonal antibodies and the immobilized monoclonal antibodies. Now you can see the test strip shown over here, on which there are 5 different areas has been shown. The number 1, a sample pad where saliva is added to the test strip. So saliva will be added over here, and then area containing mobile monoclonal antibodies attached to tiny gold particles. So the antibodies will be with the gold particles. So the antibodies are of V shape, and the part 3 test areas containing immobilized monoclonal antibodies that bind to proteins secreted by the pathogen that causes TB. Again, here will be the immobilized monoclonal antibodies. So the control area, and then there will be the area where the test strip can be held. So test strip can be held over there. So direction of flow of saliva through the test strip. So it will be in this direction from a 1 towards the 5. So in this way, the direction of flow of saliva will take place. Now, a sample of saliva is collected during this investigation. First of all, the collection of sample of saliva and put into the sample pad in the test strip. The saliva moves up the test strip through area 2. It will be, you can say it will be kept at 1 and then it will move through area 2. The mobile monoclonal antibodies are attached to tiny gold particles, and if these antibodies collect the test area 3, a gold line becomes visible on the test strips. So in this way, if gold line that becomes visible in area 4 confirms that the test strip is working and that the results are valid. So the first question in this regards is, state the name of the pathogen that causes TB or tuberculosis. As you know, 2 different types of bacteria, they can cause tuberculosis. They are Mycobacterium tuberculosis or Mycobacterium bovis. So both of they can cause the bacteria, are the bacterial pathogens which can cause the tuberculosis. So next is name the part of the monoclonal antibodies that binds to the protein from the pathogens. Part of the monoclonal antibodies, and the mono antibodies are of V-shaped, and this is the site where the uh, antibodies that binds to the protein from the pathogen. So this is the binding place. So this is called as antigen or protein binding site. So where they will attach themselves at over there. So next question is saliva is added to a test strip to test for the presence of protein secreted by the TB pathogens. Figure 3.3 is a diagram showing some of the molecules in area 3 of the test strip when a positive result for TB is obtained. It means there will be the result line. So monoclonal antibodies attached to a tiny gold particle. Protein secreted by the TB pathogen attached to the two different monoclonal antibodies. You can show this is the protein which is secreted by the patient who is suffering with tuberculosis. So these are the monoclonal antibodies attached to a tiny gold particle. So this is the tiny gold particle. And on the other hand, there are immobilized monoclonal antibodies in area 3 attached to test line. So this is the test line. They are attached on the test line. Now question is use the information so use the information in figure 3.3 to suggest and explain why this test is specific for TB. And here you can see that the test uh, which was conducted in the figure 3.3, you have seen the results. So we will understand the components first. First of all, the monoclonal antibodies, which are attached to a tiny gold particle. So these are the mobile components which can move. So the monoclonal antibody is designed to bind specifically to the protein secreted by the tuberculosis pathogen, and the gold particle is used to visualize where the binding occurs. So it is for visual activity. So the protein, second component, we can say that the protein secreted by the TB, so pathogen. So this is the target molecule. We are trying to detect if TB is present, this protein will be in the saliva. So T will be in saliva. So the third one, immobilized monoclonal antibodies uh, in area 3, which is test line, So this is another set of monoclonal antibodies also specific to the TB protein, but they are fixed in place on the test strip. So they are fixed. So now we can see the application as saliva will be used for this purpose. And then we can say that the process will take place. So these are the specific antibodies. So they are designed to bind only to the protein secreted by the TB pathogen, and they will not bind to other proteins or substance, ensuring that the test is targeting the correct molecule. So they have complementary shapes to that protein which will be secreted by the uh, pathogens. So only that protein is able to attach on the binding site, because as you know, in enzyme substrate complex, there are active sites where the only specific substrate can attach to the binding site of that specific enzyme. So same is the case over here. That is that this test is highly specific for tuberculosis, because the monoclonal antibodies are designed to bind only to the protein secreted by the TB pathogens. Other proteins will not attach themselves. They are unable to attach themselves on the binding site. So they will not bind to the other proteins or substance. They will only bind with the TB protein. So ensuring that the test is targeting the correct molecules. So in this way, only these are they have complementary shape. So they will attach only the tuberculosis. So the gold particles which are used over here, they are where the binding occurs. So they will be only for this visual purpose. So where the binding occurs. So in this way, we can say they are. This test is specific for TB. Now next question is the area 4. The area 4 contains different immobilized antibodies to those in area 3. So the mobile monoclonal antibodies bound to tiny gold particles will bind to these immobilized monoclonal antibodies in area 4. If the test has functioned correctly, a gold line will be visible in area 4, a gold line. So in this way, the gold line will be visible over there because the gold particles are attached with the monoclonal antibodies. So suggest how the structure of immobilized monoclonal antibodies in area 3 differ from the structure of the immobilized monoclonal antibodies in area 4. Again, first of all, we will discuss that this the area 4 and area 3. So different immobilized antibodies are present in area 4 contains immobilized monoclonal antibodies that are different from those in area 3, which is the test line. So binding to mobile antibodies, these antibodies in area 4 are designed to bind to the mobile monoclonal antibodies that are attached to the gold particles, regardless of whether the TB protein is present. So purpose of the control line is that it serves as a check to ensure the test is working correctly or not. And it confirms that the fluid is flowing properly through the test strip and reaching all areas. So it moves from the test area towards the other areas. And then I would like to say the difference in antibody structure, how the immobilized antibodies in area 3 differ from those in area 4. The key difference lies in their specificity. You know that area 3, which is the test line, so antibodies, so they are specific to the TB protein. They will only bind to the mobile antibody if it has captured the TB protein. So whereas the area 4, these are not specific to the TB. Area 3, specific to TB, and area 4, not specific to TB. So in this way, there is you can say they are designed the area 4, they are designed to bind to the mobile antibody directly, regardless of whether the TB protein is present or not. Whereas the area 3 special, you can say parking spot only for TB protein. So in this way, uh, we can say they are different from one another because they have complementary shape with the uh, TB protein, which is secreted by the pathogen. So in this way, the area 4 is different from the area 3. Let me uh, read this answer. Area 4 contains immobilized monoclonal antibodies that are different from those area 3, which are the test line. So the antibodies in area 4 are designed to bind to the mobile monoclonal antibodies that are attached to the gold particles regardless of whether the TB protein is present. So area 3 antibodies are specific to the TB protein, so they are designed to bind to the mobile antibody if it has captured the TB protein, because secondly, they have different structure, different protein structures. Both of they have different protein structures, so that is the reason they are meant for different purpose as well. So they are different from one another. Their function is different. They are meant for different purpose. So in this way, they have different structure. So they will be different from one another. Now we are moving towards the next question. The vaccination is another way of reducing the transmission of infectious diseases such as TB. So the BCG vaccine is used to help control the spread of TB. This vaccine contains a weakened strain of the pathogen that causes TB. So the BCG vaccine stimulates the development of antigen-specific memory T lymphocytes. Explain how memory T lymphocytes provide protection from TB in a person who has been given a BCG vaccination. Vaccination, you know, it contains the dead or nearly dead microorganisms which are injected into the body of the organisms. As you know, when a microbe enters into the body, so at that time the lymphocytes, they detect, they recognize the pathogens, and they mark them, and then they start secreting the antibodies, and then those antibodies are produced as a result of the toxins which are produced by the microorganisms. So it takes time it takes time to detect them, to recognize them, and then the production of antibodies. So when vaccination is already given to the organisms, it means the already microorganisms are in the body. So when vaccination is injected, the memory cells are also produced in the body as a result of that. So when microbes again enter into the body at that time, what happens? That is the already they are familiar. Memory cells are there, so they are very much familiar to the immune system. So you can say quickly the production of antibody starts, and this vaccination overcomes the that pathogen which has been entered into the body. So in this way, a vaccination play a very important role, that is due to memory T lymphocytes, which immediately start producing the antibodies. So in this way, vaccination is very uh, important for us. So now I would like, after this explanation, I have already written the answer. So I would like to read that. So vaccination is long-term time of effective immunity. The vaccination is the active immunity, which is a long-term immunity. So if the vaccinated individual later encounters actual TB bacteria, the memory T lymphocytes are activated more rapidly and vigorously compared to the primary response. During the primary response, it was, you can say, it was a use of span for activation of these microbes very large. So, but when the second immune response takes place as vaccination, due to vaccination, the body or the immune system was familiar with those microorganisms. So, there rapidly and vigorously, you can say, the production of antibodies takes place. So memory T lymphocytes recognize the TB antigens immediately without the need for antigen presentation and activation processes. There are more memory T cells specific to TB than there were before vaccination, leading to a quicker and more effective immune response, due to which the microorganisms are killed. So they are easily captured. So in this way, the vaccination plays a very important role. Now the next question is, the bladder in the organ in the body is to use store the urine. So when cells divide uncontrollably in the bladder, a tumor develops. And tumor is formed when there is uncontrollable division of the cells. So this can lead to bladder cancer. So this is the type of cancer. So the BCG vaccine has been used to treat bladder cancer. BCG vaccine is used for this purpose. So the BCG vaccine is introduced in the bladder The tumor cells take up the weakened pathogens in the vaccine and act as antigen-presenting cells. To name the process used by the tumor cells to take up the weakened pathogen, phagocytosis. Phagocytosis, in which the tumor cells are eaten up by the phagocytes. So the process, it is called as phagocytosis. It is the process by which the phagocytes eat up the pathogens. So this is the phagocytes. Phagocytosis suggests how antigen presentation by tumor cells stimulates an immune response that leads to the destruction of the tumor cells. So in this case, we can say that the BCG vaccine is used to treat bladder cancer by stimulating an immune response. So how it takes place, First of all, the phagocytosis, the process by which the tumor cells take up the weakened pathogens in the BCG vaccine. And then we can say the tumor cells now containing the weakened BCG bacteria act as antigen presenting cells. So the present antigens on their surface along with other molecules, the activation of T helper cells takes place. So these T helper cells become activated and stimulate beta cells to produce B cells to produce antibodies. They also activate cytotoxic T cells. So activation of cytotoxic T cells takes place. So these cytotoxic T cells become activated and directly attack and kill the tumor cells displaying the antigens. So beta cells stimulated by helper T cells produce antibodies specific to the bacterial antigens, also to the tumor. And then these antibodies can bind to the tumor cells, making them for destruction by other immune systems. So the immune system response triggered by the BCG vaccine also leads to a localized inflammation in the bladder. So the inflammation attracts immune cells to the area, further contributing to the destruction of tumor cells. So in simple terms, we can say that the BCG vaccine essentially tricks the tumor cells into displaying foreign flags on their surface. This makes the tumor cells visible to the immune system and which then attack and destroy them. So helper T cells boost the immune response by activating beta cells to produce antibodies, which can further target the tumor cells. And this is the, you can say, it works. So in this way it works. So after this explanation, I would read the tumor cells now containing the weakened BCG bacteria act as antigen representing cells. The present antigen on their surface, they will represent it, and then helper T cells recognize the antigen presented of the tumor cells. So T helper cells become activated and stimulates beta cells to produce antibodies. So they produce antibodies specific to the bacterial antigen. So these antibodies can bind to the tumor cells, making them for destruction by other immune cells. So in this way, this process takes place. So students, it was all about the question number 3. Please like, share and subscribe my channel, and thanks for watching.